Welcome back, everyone. I thought today I would make DNA simple. DNA is basically the blueprints for you. Many things that you're born with, or things that might develop later on in life, come from your DNA. So DNA comes in the shape of a double helix. That just means it has two strands that kind of twist around each other. In your DNA, you have four base pairs. Base pairs are these little colored dashes I put in this strand of DNA. You have A for adenosine, T for thymine, C for cytosine, and G for guanine. A and T and C and G will always pair up like that. If you were to try to put A and G together, you would have an instance of a square peg in a round hole. They don't fit together, it just doesn't work. So the DNA doesn't do that because it can't. However, it's important to know that in the nucleus, DNA often likes to hang out in a very unorganized way. It looks almost like a ball of yarn, and if the cell needs to divide, whether to replace old cells or maybe to fill in a cut or a scrape that you might have, it needs to condense to be more manageable for the cell. That's when you get chromosomes. In your cells, you have 23 pairs of chromosomes, 46 in total. You get one from your mom and one from your dad. In a future video, I'll discuss more about inheritance and traits like that. But for now, just know that your DNA will condense down to chromosomes, so that it's easier for the cell to divide, and each of those daughter cells to get half of your DNA. It can then duplicate, so that each of those new cells has your entire set of DNA. And if need be, those two new cells can divide, and the process continues. With RNA is where the idea of DNA gets a little bit more tricky. Occasionally, a segment of DNA can leave the nucleus. It'll then be unzipped by a protein, and another protein will come along and read one of the strands of DNA. When that happens, you'll get a strand of mRNA. The mRNA looks more similar to the partner strand to the DNA, as opposed to it being a copy of the DNA. For instance, if you were to read the strand of DNA that goes ATCG, the strand of RNA would read UAGC. The base pair uracil replaces thymine in RNA. So if ever you see the base pair thymine, you know you're working with DNA. If you see uracil, you're working with RNA. Once you have the RNA transcript that was transcribed from the DNA, it can go to a ribosome. Whether that ribosome is on the endoplasmic reticulum, or maybe it's a free ribosome in the cytoplasm of the cell. It'll be sandwiched between two parts of the ribosome. And then tRNA, transfer RNA, can come, and it can bring amino acids to the ribosome, and it can build these long chains of amino acids to make proteins. Proteins do a lot of the heavy lifting in the cell. Some proteins accelerate processes like building things or breaking things down. As I mentioned before, there's a protein involved in the unzipping of DNA, and then you have to use a protein to read the DNA. Proteins do all these crazy things, so for now, I hope this was a good little surface level exploration, my apologies, of DNA. In the future, I may go more in depth, but for now, I hope you have a wonderful morning, afternoon, or good evening.